Gosh, I don't understand the ATP production. Let me search. Oh, this one. This might help me.
Hi, my name is Francheska Millesca, and I'm here to teach you about ATP production from the complete oxidation of glucose. Let's go! The complete oxidation of glucose is a multi-process that takes place into three stages. The first stage is the glycolysis. In the cytoplasm, glucose, a 6-carbon molecule, is converted into 2 molecules of pyruvate. The net gain is 2 ATP molecules via substrate level phosphorylation and 2 NADH molecules. The second stage is the pyruvate oxidation and the citric acid cycle in the mitochondria. Pyruvate is oxidized to acetyl CoA, which enters the citric acid cycle. For each glucose molecule, this cycle produces. The third stage is the oxidative phosphorylation. This and this donate electrons to the electron transport chain. ATP is generated through the chemosymbiosis as protons flow back in the mitochondrial matrix via ATP synthase. Yields approximately 28 to 30 ATP molecule. Total ATP yield, approximately 32 ATP per molecule of glucose, varies slightly depending on subtle system. And that is all for this video. Hope you learned something today. Bye!
Oh, that's what ATP production is. What about the glycogen synthesis and degradation? Let me search. Oh, this might help me.
Hi student! I heard that you can understand glycogen synthesis and glycogen degradation, and I'm here to help you. What is glycogen synthesis?
What?
Glycogen synthesis or glycogenesis is the crucial process by which our bodies store excess glucose as glycogen to readily preserve energy. This process primarily takes in the liver and muscles. Let's break down the key steps. Glucose entering the cell is first phosphorylated by hexokinase or glucokinase, becoming glucose 6-phosphate. Glucose 6-phosphate is then rearranged into glucose 1-phosphate by phosphogluconeogenesis. This glucose 1-phosphate is then activated by UDP glucose pyrophosphorylase, which attaches a uridine diphosphate group to UDP glucose, the active form ready for glycogen synthesis. The enzyme glycogen synthase takes center stage here. The UDP portion is removed of the UDP glucose and it adds the glucose molecule from UDP glucose to the non-reducing end of the glycogen chain, forming an alpha-1,4 glycosidic bond. This process repeats lengthening the glycogen chain. As the chain grows, the branching enzyme steps in. It creates branches by forming alpha-1,6 glycosidic bonds, connecting a portion of the existing chain to a new point. Further down, this branching is key. It significantly increases the number of non-reducing ends available for both glycogen synthesis and later breakdown, making glucose readily accessible. Glycogen is primarily stored in the liver and muscles. Within the cytoplasm of the respective cells, liver glycogen maintains blood glucose levels, especially during fasting, providing energy for approximately 24 hours. Muscle glycogen serves as an energy source directly utilized during physical activity. In short, glycogenesis efficiently converts excess glucose into a highly branched glycogen molecule, providing a readily accessible energy store regulated by hormonal signals to maintain blood glucose homeostasis. The branching structure is key to its rapid mobilization when energy is needed. Glycogen degradation is the process by which glycogen is broken down into glucose, like what your ex did. Glycogen phosphorylase enzyme is the key player in glycogen degradation. It breaks the alpha-1,4 glycosidic bonds between glucose units in glycogen, releasing glucose 1-phosphate. Glycogen phosphorylase breaks from the non-reducing ends of the glycogen branches, meaning it removes glucose units one at a time. When glycogen phosphorylase reaches the branch point, it can't debreak. The debranching enzyme comes into play, transferring the 3 glucose units from the branch to a nearby linear chain. It then breaks the alpha-1 glycosidic bond, releasing the single glucose unit at the branch point. Glucose 1-phosphate, the product of glycogen phosphorylase, is converted to glucose 6-phosphate by the enzyme phospholipase. The final product of glycogen breakdown, which is glucose 6-phosphate, has different fates depending on the cell type. In muscle cells, it fuels energy production through glycolysis. Liver cells, however, can convert it into free glucose, releasing it into the bloodstream to maintain consistent blood sugar levels. Now you know what glycogen synthesis and glycogen degradation is. I hope you learned a thing and two. Adios!
Now I know. What about the gluconeogenesis? This might help me.
Hi, I'm Marianni, and I'm going to teach you about gluconeogenesis. Gluconeogenesis is the metabolic process by which organisms produce glucose from non-carbohydrate precursors, such as amino acids and glycines. This process primarily occurs in the liver and it helps maintain blood sugar levels during periods of fasting or intense exercise. The gluconeogenesis is crucial for sustaining energy levels when dietary glucose is unavailable. Function. The function of gluconeogenesis is to ensure a steady supply for glucose for energy, especially during times of fasting or low carbohydrate intake. Helps maintain blood glucose levels by synthesizing glucose from non-carbohydrate sources like lactate and amino acids. Ultimately, gluconeogenesis supports vital organs like the brain, which rely on glucose as their primary energy source. Importance. Gluconeogenesis is essential for maintaining blood glucose levels during periods of fasting, prolonged exercise, or carbohydrate restriction, ensuring a continuous energy supply. It prevents hypoglycemia by producing glucose from non-carbohydrate precursors when dietary intake is insufficient. And that's a wrap up for gluconeogenesis. I hope you learned something from this topic.
Now I understand what is gluconeogenesis. How about the terminology for glucose metabolic pathways?
Hello guys! I am Nicole without H. I am Nicole with H. We are 2 and 21. Bako yan! And we're here to help you to understand what is terminology for glucose metabolic pathways. Glucose metabolic pathways convert glucose into energy, powering cellular activities.
Understanding key terminologies is crucial for grasping these complex biological processes.
First, glycolysis. Glucose breakdown into pyruvate, releasing energy.
Second, citric acid cycle generates energy-rich molecules from pyruvate.
Third, pentose phosphate pathway produces NADPH and pentoses.
Fourth, gluconeogenesis, glucose synthesis from non-carbohydrates.
And lastly, glycogen metabolism, glucose storage and release.
Terminology in glucose metabolism is important for several reasons. 1. Clear communication. Specific terms like glycolysis and gluconeogenesis helps people to understand and talk about metabolic processes accurately. 2. Understanding pathway interactions. Terms identify key enzymes and processes like hexokinase in glycolysis and pyruvate carboxylase in gluconeogenesis. 3. Enzyme and substrate specificity. Names of enzymes and substrates reflect their functions, such as glucose 1,6-biphosphate in glycolysis. Number 4. Clinical relevance. Understanding terms can lead to misdiagnosis, like confusing hyperglycemia, high blood sugar, with hypoglycemia, low blood sugar. 5. Research and development. Proper terminology helps structure experiments and communicate findings. Number 6. Educational value. Consistent terms make teaching and learning metabolic pathways easier. Functions of glucose metabolic pathways. 1. Energy production ATP. 2. Biosynthesis fatty acid synthesis. 3. Glucose homeostasis. 4. Cellular protection oxidative stress reduction. Number 5. Signaling pathways insulin signaling cellular growth.
Bye guys! Hope you learned something in this topic.
Now I understand what are the lessons in the biochemistry, and now I'm ready for the final exam.
Bye! Thanks for watching!
Two hours later.
I mastered truffle. What about the gluconeogenesis? What about the glucogenesis?
Gluconeogenesis.
Now I know what is glucogen.
Process that takes place into three stages. And the citric acid cycle about ATP production. The first stage is the glycogenesis. What? Glycogen. Break down the key steps.
Guys, I am going to eat. I am going to eat.
Here you go.
Hello guys. Hello guys.
Arayo ka mas luwa. Arayuan ka mo.
Hello guys.
Kaya magkaram.
Hello guys.
Bako yan! Bako yan! Go! 3, go! Seconds.
Second.